Welcome, this is Ad Emmen reporting live from the Super Day event in Amsterdam, organized by SURFsara. And we are talking here with Jan Marrink, who's head of the Molecular Dynamics department at the University of Groningen. And he just gave a keynote presentation here. So welcome.
Thank you very much.
Okay. So can you tell a little bit about what you do?
Yes, so the work that we do in Groningen in the Molecular Dynamics group is to use computers, big computers, to simulate the interaction of molecules, and in particular the molecules that build cell membranes. So we're interested in understanding how cellular membranes are organized, how the lipids and the proteins that make up these membranes are self-organized. So this is a very complex matter because cell membranes are composed of hundreds of different lipid-type molecules crowded with proteins, and they together make or give rise to the fundamental processes that govern the functioning of cells.
You showed some examples of cell membranes, and they're usually complex, I must say. I mean, why are they so complex?
Well, that's actually a very good question. Why are they complex, and why are they so complex? Because if you think of the basic functionality of a cell membrane, it's just to provide a barrier between the inside and the outside of a cell. And for that, you only need lipid molecules that form a bilayer, and any lipid molecule can do that. So still there is hundreds of different types of lipids, and we are only at the beginning of trying to understand why there is such a need for such a large diversity. And it seems that many lipids specifically bind to proteins and are able to bring proteins together, and not just any proteins, but proteins that also need to function together. So the lipids seem to have a nice steering role in governing the organization of the cell membrane.
So in the old days, you would take a cell and do something with it, prepare it, and put it under a microscope and look at the structure. But that's not the way that you do it today. So how do
no, so you can still do that, and of course many experimental colleagues, they look at membranes in that way. But the main problem is that the resolution that you can attain with traditional microscopic techniques is very limited. So you can not see the individual lipids and the proteins that are the basic building blocks of the membrane. So what we do is then what we call computational microscopy, where you actually simulate the interaction between all the components in a virtual reality. But then the advantage is that you have the resolution down to the atomistic, the individual atoms of the molecules, and also at time scales that are from the picoseconds to the nanoseconds and the microseconds, you can resolve the dynamics. So this is the big advantage of the computational microscopy approach over traditional experimental microscopy.
Yeah. So you build the model and put it in a computer and then do the calculations and make a nice visualization so that you can see what really is happening.
Yeah, so what we actually do is we solve the equations of motions as already formulated by Newton many centuries ago. So all the atoms, they exert forces on each other, and they give rise to that the molecules start moving around. And so this is we can simulate if we have enough computational resources, and so that we can visualize what will happen to the system over a period of time. And this is usually microseconds up to milliseconds of time that we can simulate these systems.
You were mentioning computational resources, but it's not something you can do at home, I guess, with your home PC or...
No, only a very small, limited part of a cell membrane you could do at a laptop at home. No, if you really want to go to larger systems and follow the dynamics for an interesting amount of time, you need the biggest supercomputers that you can lay your hands on, basically.
Why does the model require so much computer time?
Well, basically because you have to compute the forces between all the particles that are in your system. So these can be millions and millions of atoms. And also you have to do this you solve this iteratively. So you do one time step at a time. And the time steps that you can take between different snapshots of your system are of the order of a couple of femtoseconds. And to get to microseconds, you have to have millions and millions of these sequential time steps that you have to calculate. And that is just, a huge computational effort.
Mm-hmm. And you're currently using the supercomputers at SURFsara here in Amsterdam.
Yes.
Are you also using other supercomputers, or is it the main one?
Mainly so we have our own local supercomputers at the University of Groningen. But then at the national level, we rely on the SURFsara supercomputers. Sometimes we're also involved in European projects, but the bulk of our simulations is done here in Amsterdam.
So on the European scale, they're working on an exascale supercomputer somewhere in 2020, 20, whatever. Would it solve all your computational problems?
I wouldn't say it would solve them, but it would definitely help. Currently, we're always facing the limit of the computational resources that we have. So if we can really get to the exascale, then this would open up a whole new box of simulations and systems that we can look at and processes that we can study. Because still we are limited in time scales that are far away from a lot of cellular processes that take place on the time scales of seconds or more that we currently cannot reach with the current computational power.
Yeah, at the presentation you are mentioning that you, well, not in the end, but in some time you would like to have a simulation or a model of the complete cell, one complete cell. So when do you expect that?
Yeah, that is something that this is our goal for, say, in about 10 years. We hope that we can construct a model of a full biological cell, so including the cell membrane with all the different lipids and proteins that are found there, but also the interior of the cell, the cytoplasm, some of the internal organelles. So really get to a complete cell model where you still have access to the molecular resolution that gives rise to the whole functioning of the cell. And I think this is required because all the components are interconnected with each other. So of course you can study processes in isolation, but at the end the cell functions because all the processes are connected. And this eventually we hope to also capture in our simulation models.
I'm looking forward to the presentation in 10 years' time about a complete
That's
cell.
my promise now, and I will show you a nice movie of a complete cell simulation.
Okay, thank you very much for this interview.
My pleasure.
For Primeur Magazine, this was Adam reporting.